Imagine a world free from genetic diseases. Imagine a future where we can enhance our abilities and resilience. This isn't science fiction, this is the promise of CRISPR, a revolutionary gene editing technology. CRISPR gives us the power to rewrite the code of life, to correct genetic defects, and to enhance desirable traits. CRISPR stands for clustered regularly interspaced short palindromic repeats. It's a mouthful, but the concept is simple. CRISPR is like a pair of molecular scissors that can precisely cut DNA at specific locations. This precision is what makes CRISPR so powerful, and why it has taken the scientific world by storm. The implications of this technology are vast and profound, touching every aspect of our lives. From curing diseases to enhancing crops, from reshaping ecosystems to redefining human potential, CRISPR has the power to reshape our world. But with great power comes great responsibility. As we stand at the cusp of this genetic revolution, we must proceed with caution, engaging in open dialogue and ethical considerations to ensure that this powerful tool is used for the betterment of humanity and our planet. At its core, CRISPR is an elegant system borrowed from bacteria. Bacteria use CRISPR as an immune system to defend against invading viruses. Scientists have harnessed this natural system to create a powerful gene editing tool. The CRISPR system consists of two main components, a guide RNA and a CA's protein. The guide RNA is like a homing beacon, designed to recognize and bind to a specific DNA sequence. The CA's protein acts as the molecular scissors, cutting the DNA at the location specified by the guide RNA. Once the DNA is cut, the cell's natural repair mechanisms kick in. Scientists can exploit these repair mechanisms to introduce changes to the DNA sequence. This could involve disabling a harmful gene, correcting a mutation, or even inserting a new gene altogether. The precision of CRISPR is remarkable. It's like having a word processor for DNA, allowing scientists to target and modify specific genes with incredible accuracy. This level of precision opens up a world of possibilities for treating genetic diseases and enhancing desirable traits. Genetic diseases have plagued humanity for millennia, causing untold suffering and hardship. CRISPR offers a glimmer of hope for millions affected by these debilitating conditions. Imagine a future where we can cure cystic fibrosis, sickle cell anemia, and Huntington's diseases caused by single-gene mutations. CRISPR has the potential to correct these mutations at their source, offering a permanent cure. Clinical trials are already underway, testing the efficacy of CRISPR in treating various genetic disorders. Early results are promising, demonstrating the potential of this technology to alleviate human suffering. But the promise of CRISPR goes beyond single-gene disorders. Scientists are exploring its use in treating complex diseases like cancer, Alzheimer's, and heart disease, which involve multiple genes and environmental factors. The ability to edit our genes raises profound questions about what it means to be human. Could we use CRISPR to enhance our abilities, to make us stronger, smarter, or more resistant to disease? The possibilities are both exhilarating and unsettling. Imagine a world where parents could choose their children's traits, enhancing their intelligence, athleticism, or even their physical appearance. While such scenarios raise ethical concerns, the potential benefits are undeniable. CRISPR could help us create a healthier, more resilient human race, better equipped to face the challenges of the future. However, we must tread carefully, engaging in thoughtful discussions about the ethical implications of genetic enhancement. The decisions we make today will have far-reaching consequences for generations to come. Section 5. A world transformed CRISPR in agriculture and beyond. The transformative potential of CRISPR extends far beyond human health. It has the power to revolutionize agriculture, reshape ecosystems, and even create new forms of life. Imagine crops engineered to be more resistant to pests, diseases, and harsh environmental conditions. CRISPR could help us feed a growing global population and reduce our reliance on harmful pesticides. Scientists are already using CRISPR to develop crops with enhanced nutritional value, longer shelf life, and improved drought tolerance. These advancements could have a profound impact on global food security. Beyond agriculture, CRISPR is being used to develop new biofuels, create disease-resistant livestock, and even control populations of disease-carrying insects. The possibilities are limited only by our imagination. Section 6. Ethical Crossroads. Navigating the CRISPR Revolution. The power of CRISPR to alter the very fabric of life raises profound ethical questions. As we venture into this uncharted territory, we must proceed with caution and engage in thoughtful deliberation. One of the most pressing concerns is the potential for unintended consequences. Editing one gene could have unforeseen ripple effects on other genes or even entire ecosystems. We must be mindful of the delicate balance of nature and avoid unintended harm. Another concern is the potential for exacerbating existing social inequalities. Access to CRISPR-based therapies and enhancements could be limited to the wealthy, creating a genetic divide between the haves and have-nots. We must ensure that the benefits of this technology are shared equitably. Perhaps the most profound ethical dilemma is the potential to alter the human germline, the DNA passed down from one generation to the next. Such changes would be permanent and irreversible, impacting not only the individuals treated, but also their descendants. Section 7. The Uncertain Future. A World Shaped by CRISPR. The CRISPR revolution is still in its infancy, but its impact on our world is already being felt. As this technology continues to advance, it will undoubtedly shape the future of humanity in profound ways. The potential benefits of CRISPR are undeniable. It offers hope for curing diseases, enhancing human capabilities, and addressing some of the world's most pressing challenges. But the risks are equally real. We must proceed with caution, guided by ethical considerations and a deep respect for the sanctity of life. The decisions we make today will have far-reaching consequences for generations to come. The future with CRISPR is uncertain, but one thing is clear: this technology has the power to reshape our world in ways we can only begin to imagine. Section 8. Balancing Innovation and Responsibility. The rapid pace of CRISPR research and development presents us with a critical challenge: how to balance the pursuit of scientific innovation with the responsibility to use this powerful technology wisely and ethically. Open and transparent dialogue is crucial. Scientists, ethicists, policymakers, and the public must engage in thoughtful discussions about the potential benefits and risks of CRISPR. We need to establish clear ethical guidelines and regulations to govern the use of this technology. Education is also paramount. We need to educate ourselves and future generations about the capabilities and limitations of CRISPR. Informed citizens are better equipped to make responsible decisions about this powerful technology. Ultimately, the future of CRISPR lies in our hands. It is up to us to ensure that this revolutionary technology is used for the betterment of humanity and our planet. Section 9. CRISPR and the Definition of Humanity. The ability to edit our genes forces us to confront fundamental questions about what it means to be human. If we can change our genes, are we still human in the same way? Some argue that genetic enhancement undermines our humanity, turning us into products of technology rather than creations of nature. Others contend that our ability to shape our own evolution is a natural progression of human ingenuity. The debate over CRISPR challenges us to re-examine our values and beliefs about the nature of life itself. It forces us to confront our fears and aspirations as we grapple with the implications of this transformative technology. As we navigate this uncharted territory, we must be guided by wisdom, compassion, and a deep respect for the diversity of life on Earth. Section 10. The Need for Global Dialogue and Regulation. The CRISPR revolution knows no borders. This technology has the potential to impact every country and every culture on Earth. As such, it is imperative that we engage in a global dialogue about its ethical, social, and environmental implications. International cooperation is essential to ensure that CRISPR is used responsibly and for the benefit of all humankind. We need to establish global norms and standards to prevent the misuse of this powerful technology. A global governance framework for CRISPR should address issues such as equitable access to CRISPR-based therapies and enhancements, prevention of genetic discrimination, protection of human dignity and genetic diversity, regulation of germline editing, and environmental risk assessment and management. Section 11. Conclusion. CRISPR, a legacy for future generations. The discovery of CRISPR is a watershed moment in human history. It has given us the power to rewrite the code of life, to correct genetic defects, and to enhance desirable traits. The potential benefits are vast, but so are the risks. As we stand at this crossroads, we must proceed with caution, guided by ethical considerations and a deep respect for life. The decisions we make today will have far-reaching consequences for generations to come. The CRISPR revolution is not just about science and technology, it's about our values, our beliefs, and our vision for the future. It's about what kind of world we want to create for ourselves and for future generations. We must use this powerful tool wisely, ensuring that it is used for the betterment of humanity and our planet. The legacy of CRISPR will be determined by the choices we make today. Let us choose wisely.